Okay, we're just going to fly through high points of the next two chapters since we've changed how the test will be run. So this chapter covers amino acids, peptides, and proteins. And you can see the important sections here. I won't even discuss sequencing a peptide or peptide synthesis or protein function. So amino acids are connected together through a amide bond. In biology, this is referred to as a peptide bond. And you notice that we have these amino groups and these acid groups. So if we link these together to make an amide bond, that leaves a free carboxylic acid that you could link another amino group to. So you could link these together in very long chains. We would refer to these as polymers generally, where this is referred to as the monomer, and this is the polymer. In this case, we're making polymers of amino acids, again, linked through an amide bond, or in biological terms, peptide bonds. Kind of a side note, these amide bonds are also found in Kevlar, so they tend to be very strong bonds. So if we look at the amino acids and we draw them in a Fisher projection like we did for sugars, you notice that the nitrogen is off to the left. And so these amino acids are typically referred to as L amino acids. So nature prefers D sugars and L amino acids. Again, this D and L has nothing to do with the actually R and S configuration. As we actually see down here at the bottom, you can see that even though all three of these amino acids are referred to as L amino acids, cysteine actually has an R configuration. So we'll see that one amino acid, glycine, doesn't even have a stereocenter, and then cysteine actually has the R configuration, even though they're all considered L amino acids. And the reason that cysteine has a higher, or sorry, R configuration is that as we number out, you get to the oxygen and the sulfur at position 2, and sulfur wins out over oxygen. And this is the only amino acid that is the case. So cysteine has an R configuration, even though it is considered L. Again, L comes from the Fisher projection with the amino group pointed out to the left. Just like we saw in sugars, if we happen to have that last chiral center pointed to the left, we refer to it as an L sugar. Again, nature prefers the D sugars and L amino acids. So we can classify the amino acids based on their side chain nature. So if you haven't figured out, all amino acids contain an amino group and a carboxylic acid group. They are connected to the same carbon, referred to as the alpha carbon, and then they have a side chain. This side chain is referred to as R, or sometimes a residue. And so this R group can be nonpolar. The R group can actually be polar and neutral, meaning that the side chain contains an oxygen or sulfur, but no charge. So the side chain is able to participate in hydrogen bonding or has some polarity to it, but it doesn't have a charge. And then we have the polar acidic amino acids. They contain a carboxylate group on their side chain. So not only do they have a carboxylic acid that makes them an amino acid, their side chain also contains a carboxylic acid. And then we have the polar basic groups. These are the groups that like pumpkin spice lattes, and they contain an amino group with a positive charge in the side. And anyway, this is the general structure. They have the amino group, they have the carboxylic acid group, the alpha carbon, and the side chain. And then they'll have an H off of that alpha carbon as well, for most cases. So if we look at just side chains, and you'll notice that I'm moving from a couple different representations here. I'll show you my preferred representation of these. Right now, we're just trying to get an idea about how the amino acids look and their side chain. So the nonpolar side chains tend to be those that are just hydrophobic, have no polarity to them, just all carbons. And so we have alanine and glycine and isoleucine, and then we have leucine. So we have leucine and then this isomer. Notice that they're both butyl side groups. This is actually a sec-butyl side group, and this is an isobutyl side group. So leucines have these butyl side groups. Isoleucine is a sec-butyl, and leucine is an isobutyl. Don't ask me why those don't match up. That is just the way it is. Methionine, even though it does have a sulfur here, it is a sulfide. It's got carbons on either side of that sulfur. So it's a lot like an ether, just not very polar. So it's classified as a nonpolar side chain. And then, of course, large aromatic groups, the phenylalanine. A lot of times people forget this CH2 here. So it's actually a benzyl group connected to that alpha carbon. You don't want to draw the aromatic ring connected directly to that alpha carbon. That is not one of the canonical or the 20 standard amino acids. We see an interesting one. This is proline. It is unique in that the side chain actually back bonds to the nitrogen and forms a ring. It's the only amino acid to do so. The rest of them just have their side chain dangling off the alpha carbon. Proline actually forms this 5-membered ring by having the side chain back bond into the amino group. And then finally we have valine. And if we drew it out, it's actually a isopropyl group, but it looks like a V there. Oh, sorry, I forgot tryptophan. Let's not forget tryptophan right here. This is supposedly what makes you sleepy when you eat lots of turkey. That's probably more of an urban legend, but this molecule here is turned into serotonin, which is the feel-good neurotransmitter. Okay, let's look at the polar but neutral side chains. So these are all going to have atoms in the side chain that convey some polarity to them. So asparagine actually has an amide in the side chain. Cysteine, like I said, different than methionine, actually has an SH there. And so this SH gives it a little bit more polarity, and so it's considered a polar but neutral. Glutamine right here, which is related to asparagine. The difference is it just has one additional carbon. Contains an amide bond, again, adding some polarity to the side chain. We have serine over here, and then we have serine, and then if we add a carbon off to this group here, we have threonine. And then we have something that looks like it's related to phenylalanine, but it has an OH, so it's a phenol, and that is referred to as tyrosine. And then finally, we have these polar and charged side chains. We have two that have carboxylic acids. Again, they're related by having this carboxylic acid and only differ by having one additional carbon. So aspartic acid, and then glutamic acid. So glutamic acid just has that additional carbon there. Otherwise, they're identical. And so those are the two amino acids that have acidic side chains. And then we have basic side chains. And they kind of vary in structure. Down here, we've got lysine that contains just a normal amine. It's actually got a butyl group contained to an amine, and that is lysine. We have this arginine here that then contains this interesting motif right here known as a guanidine. It can still act as a base, and so it is classified in the basic amino acids. And then we have this cyclic histidine right here, and it also has a nitrogen that can act as a base. So those are all basic amino acids. Again, what they have in common is the nitrogen that can pick up a protein. If you're suffering from allergies right now, You're suffering from histamine, which is actually this molecule right here that's circled, and it differs from histidine in that it's been decarboxylated. So if you take an antihistamine, you're taking something that blocks the release of this molecule in the circle right there. Okay, like I said, there's a better way to draw these out, and I learned this in graduate school, and I'm really a fan of it. There's many different reasons to draw it out in this way, but what you do is you draw the nitrogen off to the left, you draw the little valley here, and then the carboxylic acid off to the right. Now you may notice here something different from the previous structures in that there's no charges on what I'm drawing here. And I'll talk about that in just a second. Just a little heads up, though, this is actually not the correct charged form of amino acids. They never actually exist in this neutral looking form, but just a lot prettier to look at this way. So how I've arranged them is there's some logic here. So I didn't necessarily arrange them by polarity or charge side chain. I arranged them related by their structural features. So the first row here, what we've got is just plain old carbon side chains. And then you say, wait, glycine doesn't even have a carbon side chain. And you're right. But these are just the simple hydrocarbon side chain. So we got glycine. As I mentioned earlier, it is the one amino acid that is not chiral because it's got two H's off of its alpha carbon. Then we get to the simplest amino acid, which is alanine. And notice when you're an L amino acid, that R group will always be back when you draw it in this form. If you draw the nitrogen off to the left with this little valley right here, then the R group will always be pointed back in natural amino acids, in the L amino acids. So we've got alanine, then we add another carbon on there, and so we get to valine. Then what we do is we essentially just add an additional carbon in here. So if you look, that looks a lot like valine, but we've added an additional carbon. And so that is referred to as leucine. Again, it's an isobutyl group that we have attached to the R group, but that is leucine. And then isoleucine is the sec-butyl group attached. Again, this first row just contains all carbon-hydrogen bonds. Now we go down to the next one, and they're all rings in the side chain. And so if we go, we have phenylalanine that contains this benzyl group. We modify that benzyl group and put an OH and make it a phenol. We get tyrosine. And then we get into kind of the weirder ones. We get histidine, which contains this imidazole side chain. And again, it's classified as a basic amino acid because it has a lone pair off that nitrogen that can act as a base. We have tryptophan here. It's not classified as a basic amino acid because that lone pair right there is actually involved in resonance that makes that ring aromatic. This lone pair on histidine actually is involved in resonance, but this lone pair is actually found in an sp2 orbital. So we've talked about this before. Those in the sp2 orbital can actually act as a base. And then we always have the oddball that doesn't quite fit in. It still has a ring, but it's not an aromatic ring. It's just a heterocyclic ring, and that is proline. Again, still fits in the rings. It's just not a ring right off the side chain. It is a ring that is formed by bonding with the amino group of the amino acid. Then we go down to the next one, and these are all amino acids that contain a heteroatom, meaning atoms other than carbon and hydrogen, and they don't have a ring. And so the simplest one is going to be serine, and then threonine, which is related. It just has an additional methyl group that does actually contain or make another chiral center there. So you notice that there's actually two amino acids, isoleucine and threonine, that both have additional chiral centers in their side chains. In nature, isoleucine, the side chain tends to be in the S configuration. And you can kind of remember that by its iso S there. And then threonine, the side chain tends to be R, and you can kind of remember that it's ther. Anyway, so essentially the second or third letter in here tells you what configuration the side chain is in a typical natural amino acid. Now, if we switch out that oxygen for a sulfur, we end up with cysteine as the amino acid. Now, we have methionine. It differs from cysteine in two different ways. It has an additional carbon here, and it's a sulfide. So instead of having an H there, it actually has a CH3. So the chain is a little bit longer, and it has a CH3 out there. And then we get to lysine, and like I said, it contains 4 carbons, so a butyl group. And then you're just connected to an amine. So these are all just side chains that have atoms other than carbon and hydrogen without rings. And then we get down to the final row, and we have all those side chains that contain C double bond O's. And of course, the one weird one, a C double bond N. So they contain a C double bond to a heteroatom. So we have aspartic acid, which is an acid, and its relative, the amide, which is asparagine. So you can kind of remember the A's, aspartic acid and asparagine have just 1 carbon, and the acid has an acid, and the asparagine is the amide bond. Then we have these relatives to aspartic acid, has 1 additional carbon. So it's referred to as glutamic acid. So G comes after A, so glutamic acid comes after aspartic acid, and it has an additional carbon there. And then, of course, we have its relative, same number carbons as glutamic acid, but it has an amide instead of a carboxylic acid, and has a similar sounding name, glutamine. So again, aspartic acid and asparagine are related, just differ in the functional group. Really, they only differ in having an oxygen or a nitrogen. And then glutamic acid and glutamine differ at the same point. And then you just have arginine, which is just really kind of an odd one. It's got 3 carbons here, bound to a nitrogen. So it's actually not derived from lysine. It actually has 1 less carbon, and then it comes to an amine. It's actually derived from an amino acid known as ornithine. This nitrogen here is bound to this carbon. Carbon has 4 bonds, and this carbon is surrounded by 3 nitrogens. So carbon with 4 bonds surrounded by 3 atoms must have a double bond to one of those nitrogens, and you can see it does. So that's how I like to lay them out. You know, it suggests that you learn their names, their 3-letter codes, and even their 1-letter codes, as well as their structures. You'll see this again and be expected to know this in your biochemistry course. If we were taking a normal test, I'd give you a chance to draw those out and get some points, but we're not doing that. So the names of the 20 amino acids are not systematic, meaning they don't have a pattern. It is really just memorization. And they have a 3-letter code, which is somewhat related to the name, and then they have a 1-letter code. Now, there's 20 amino acids. Not all of them start with different letters, and so some of them have 1-letter codes that don't necessarily match up to their name. So if only 1 amino acid begins with that letter, such as cysteine, histidine, isoleucine, methionine, serine, and valine, then that is used. So cysteine has a C, and histidine has an H, and isoleucine has an I, methionine M, and serine S, and valine V. But now there's some that have more than 1 amino acid. So alanine and arginine and asparagine, or aspartic acid, sorry. And yeah, arginine, asparagine, aspartic acid, and alanine all start with an A. So then the question is, well, who gets the A? And it typically is the one that's most common. So alanine gets the A, glycine gets the G, leucine gets the L, proline gets the P, and threonine gets the T. So what do you do with phenylalanine that starts with a P? If P's already used up, well, you kind of used phonetic. So phenylalanine as if it started with an F. Arginine, that's the pirate's favorite amino acid, arginine. So it starts with an R. And then tyrosine, yrosine. So tryptophan has a double ring, and hence it's a W. I like it better that tryptophan is like tungsten, and tungsten has the atomic symbol W. So I think of tryptophan as tungsten with this atomic symbol or one letter code of W. And then amides have letters from the middle of the alphabet. So glutamine is cutamine. That's how I remember it. And then asparagine contains an N. And this gets worse and worse as you try to make sense of it as you go down. And then the acids end in D or E. So the smallest first, aspartic acid, and then glutamic acid, E. Sure, okay. So since there's 20 amino acids, there are 3 letters that aren't used. So Jock's buzz is how you can remember that, or buzz jocks, or jocks getting buzzes. And so you can spell your name in amino acids unless you had a Joxbuz in your name. But not all is lost because all the other 6 letters are used up. So B is generic letter if you have asparagine or aspartic acid. Z is the generic letter if you have glutamine or glutamic acid. Leucine and isoleucine, again, similar in structure. So generically, they're referred to as J. If you don't know the amino acid, it is X. And then, as I mentioned earlier, ornithine has 3 carbons here. Lysine actually has 4. So ornithine is a common amino, well, I shouldn't say it's an amino acid that we find in proteins, but it is a common biological amino acid, and this is what we use to make arginine. Now, we have found organisms that include a 21st amino acid known as selenocysteine. We also have selenocysteine, but we include this in our proteins post synthesis of the proteins, but there are organisms that actually include selenocysteine directly at the ribosome. But anyway, it's got a code of U. So when we talk about these for you nutrition majors, some of them we refer to as essential amino acids. These are essential in the diet because we can't make them. And so you can see the essential amino acids listed up there. And then we have amino acids that we can make, which are the rest. So some of these are required in the diets of children. And if you don't eat a well-rounded diet, then you may be deficient. So now if you are not a vegetarian, or what I refer to as a secondary vegetarian, meaning you eat the animals that eat the plants, then you're not going to be missing out on any amino acids. If you happen to live off of wheat, rice, and oats, you're going to be lacking lysine. Corn, you're going to be missing lysine and tryptophan. And so you can see if you live a vegetarian or vegan lifestyle, it's good to and important to eat a well-balanced diet, because if you just live off some of these, you will be lacking in some amino acids that can cause a bunch of health issues. Okay, so as I mentioned, when I drew out the amino acids on my chart there, I drew it in a structure that's not really chemically correct. I drew it in a neutral form, and that's just to make it easier to look at. So the key for the true form that amino acid exists in depends on the pH. But before we get to that, we'll actually just look and see that an amino acid contains an amine group and an acid group. Well, an amine group is a base, and we have an acid group in the same molecule. So what really should happen is a proton transfer from the acid to the base. And so what we end up with is a salt. And so typically amino acids exist in this salt form where the amine is protonated and the carboxylic acid is deprotonated. exists as a carboxylate. And in this case here, in this amino acid, we have opposite but equal charges, and so we refer to that as a Zwitter ion, or the Zwitter ionic form. So overall, the molecule has no charge, it's neutral, but the molecule does have charged atoms. They just happen to cancel each other out. So amino acids really don't exist in a neutral form. They exist in this charged, but often Zwitter ionic form. Now, not all amino acids will exist this way because, as we talked about, some of them have side chains that can be polarized. There's 5 of those, or sorry, protonated or deprotonated. And so those are a special case, but for the other 15, they tend to exist in this Zwitter ionic form. But really the form that they exist in depends on the pH. So if you eat protein and it goes into your stomach, what really happens is the amino acid tends to be positively charged because everything that can be protonated will be protonated, meaning the amino group will be protonated, and in your stomach, the carboxylic acid actually will be protonated as well. So at low pH, everyone that can have an H does, and your amino acids tend to be positive. And that kind of makes sense because we're adding H pluses. So we're in an environment with lots of H plus, so our molecule also has a overall plus charge. Now, if we increase the pH, meaning we start to add base, we're going to start pulling protons off of the most acidic spots. And the most acidic spots is the carboxylic acid. That's why we call it an acid. So as we start to increase the pH or add a base or get to a higher pH, we tend to deprotonate the carboxylic acid, and this is where we get to our Zwitter ionic form. And then if we increase more and more basic, like you're trying to dispose of a body by dissolving it in sodium hydroxide, eventually you would deprotonate the amine part as well. So at high pHs, the molecule tends to be negatively charged, which makes sense because we're in the presence of OH minus. We've stripped away any positives there, and we've converted everything into O minuses. So those are the real forms of the amino acids. They're either overall positive, the Zwitter ionic form, or this negative form. And of course, that's the simplification because there's 5 amino acids that have a side chain that can be protonated or deprotonated. But for the other 15, you can identify their form by looking at the conditions. So if we just take the Zwitter ionic form here and we protonate it, we would expect the amino acid to be overall positive. So again, everyone that can pick up a proton does. This guy can't pick up any more protons. He's used up all his electrons, but the carboxylate can get protonated to a carboxylic acid. In basic solution, if we start with the Zwitter ionic form, once we add the base, we're going to start deprotonating every place that we can. We can't deprotonate at the carboxylate since it no longer has an H, so we would deprotonate the amine. And so again, in basic conditions, amino acids tend to be negatively charged, and in acidic, positively charged. Okay. I'm just going to pause here for a second. You'll look at this in depth in biochem. In short, it's just the life of amino acid as we change the pH. So at a low pH here, again, everything is going to be protonated that can be protonated. So the molecule would have an overall positive. And then as we add OH minus, that's going to cause the pH to increase. And so as we start to increase the pH and add OH minus, well, that OH minus is going to start to deprotonate the most acidic proton, which is the H off the carboxylic acid. And at some point, you'll get to a mixture where you have half protonated, half deprotonated. And that is the definition of the pKa of this carboxylic acid here. So the pKa of the carboxylic acid is 2.34. Now that is more acidic than a typical carboxylic acid, which is closer to 5. And the reason that amino acid carboxylic acids are more acidic is due to the amine group pulling electrons away, therefore making it more acidic. So we have this 50-50 mixture, and then as we keep adding base, eventually we get to the point where all of the acid has been deprotonated. And once we get to that point, we get the Zwitter ionic, and the Zwitter ion, and we refer to this as the isoelectric point, when the overall amino acid has a neutral charge, even though individual atoms are charged. So this has an overall charge of 0, even though we can see that the amino acids have charges. And then as we keep adding base, we're going to start pulling the protons off of the next most acidic proton, which is actually off of the nitrogen. And we'll get to a point where half of them have been deprotonated and half still have that. And again, that is the pKa of an amine, which is near 10. So for carboxylic acids, amino acids, a good rule of thumb is that the carboxylic acid is near 2, in between 2 and 3. And the amine group, pKa, is near 10. And this all has to do with the Henderson-Hasselbalch equation, which we're just going to conveniently skip over. So like I said, this can be more complex for the 5 amino acids that have either acidic side chains or basic side chains. So if we actually take a look at histidine, which is one of our basic side chains, we do the same thing. We actually walk through, we start at the most acidic state, and everyone that can have a proton does. So the carboxylic acid's protonated, the amine is protonated, and the amine, or the nitrogen in the side chain, is protonated. So the imidazole ring is actually protonated. Then we increase the pH and we start to deprotonate the most acidic. Well, the most acidic proton's always going to be the acid. That's why we call it an acid. Then as we increase, the next most acidic proton is off of the aromatic ring. And so we would deprotonate that around pH of 8. And then again, if we got up to a pH of 12, then everyone would be deprotonated. So you can see that this molecule goes from a +2 to an overall +1 to an overall 0 to a negative charge. So this is its Zwitter ionic form right here. But to get to that, we actually had to deprotonate twice. Okay. That is the general structure of amino acids. Now, how do we make them? Well, this is really cool because we can make them using chemistry that we've seen before, which is cyanohydrin and imine chemistry. So if you take an aldehyde and you mix it with ammonia and HCN, you end up with this cyanoamine here. And when you hydrolyze that, this, of course, turns into a carboxylic acid, and lo and behold, you have a amino acid. So how does this work? Well, first of all, if we take ammonia and mix it with the aldehyde, we get the amine. When we take the imine and mix it with cyanide with a little bit of acid, we protonate the imine, and then the cyanide attacks just like it would in a carbonyl, and you end up with this aminonitrile. And again, we would hydrolyze this just like we saw in the carboxylic acid chapter, and that would make a amino acid. That is known as the Strecker amino acid synthesis. Pretty slick. And so you could make different amino acids depending on your choice of the R group here. So if you change your R group and you have this aldehyde, you could make all 20 amino acids just by proper selection of that R group. So we can also make amino acids by using the Helvell-Hart-Zielinski reaction, which we learned back in the carboxylic acid chapter. If you take a carboxylic acid and you take it under these conditions, Br2, PBr3, remember the first change that we make is we make that into a Br, which allows us to form an enol at this alpha carbon. That enol of that alpha carbon then attacks the bromine. And before we add water, what we end up with is an alpha bromo acyl bromide. And then when we work it up with water, bromine is converted into an OH. And if you look at that, we now have a alpha bromo carboxylic acid. So we have an electrophile at that alpha position, and what we need to do is just do an SN2 reaction to kick off that bromine, and lo and behold, instead of an alpha bromo, we have an alpha amino group. And this reaction here would actually give us a mixture of R and S at that position. So when we go to make our leucine in this example, we would get a mixture of R and S there. Now, if you found some way just to make the R here, then you would end up with just the S there. Since it's an SN2 reaction, it would invert it. But most of these examples that we're going to look at are not selective. You'd get a mixture of enantiomers of your amino acids. So we've seen revisiting of the imine and essentially cyanohydrin chemistry. We've now seen the Helmholtz-Zielinski reaction. Oh my hell. Hell means light in German, so it's not as bad as you think. So let's look at some other chemistry. This is actually more recent chemistry that we learned a lot about in the enolate chapter. If you take this diethyl acetomalonate right here, okay, this is very similar to malonic ester that we saw before. And malonic esters were used to essentially undergo a transformation where you made a carboxylic acid and then you had some R group off of here. Well, this would be pretty slick if I could append a nitrogen off of that alpha group. Then when I underwent this change, what I've done is I now have my my nitrogen at the alpha carbon. I've made my carboxylic acid, and then I'm simply adding my R group on there. So that's what this diethyl acetomalonate does. So we actually run the reaction just like we would in a malonic ester. We deprotonate at the alpha position, make an enolate. That enolate would then attack a alkyl halide, kicking off the bromine, and you end up with this big, scary looking molecule that when we treat it with acid does a bunch of different reactions. First of all, it's going to convert each one of these esters into a carboxylic acid, as well as this ester right here into a carboxylic acid. It's also going to hydrolyze this amide into an amine. So essentially when you treat it with H3O+, you're going to hydrolyze the esters and the amides. And so after just this step here, what we would expect is C, carboxylic acid off there, carboxylic acid off there. Ooh, I don't have green, so we will just go to carbon, and then you'd have a carboxylic acid off of here, and then you would have an amine off of here. So that's what you get just after you hydrolyze everything. And then the heat, what it does is it actually does the decarboxylation. So one of these is going to leave as CO2, and you end up with an amino acid. Awesome, chemistry that we've seen before, essentially just a modified malonic ester synthesis. Now this one's very similar to the one before. Actually, this is a phthalimide that we saw in the Gabriel amine synthesis. So this is an N-thalidomidomalonic ester synthesis. In essence, this is just a protecting group for the nitrogen in the side chain. This is just a protecting group for the amino group in the side chain. And the reason for that is we just can't have free nitrogens because they end up being good nucleophiles. So this reaction is the same. The base is just going to come deprotonate that alpha carbon. The alpha carbon is going to do an SN2 on that substrate there. We're going to end up with an ester here, an ester here, and this imide, amide looking group. It's known as an imide. And we're going to hydrolyze it with acid. So again, this turns into a carboxylic acid, this turns into a carboxylic acid, and this would just turn into an amine. And then finally, in heat, we're going to lose one of our CO2s as gas. And again, we've made a amino acid. Again, very similar to the previous one. It's just that the protecting group that we're using on the nitrogen is different. This one's not as atom economical because you can see the protecting group is much, much larger. But we can make this one. It's actually a modified Gabriel amine synthesis. What you do is you take your malonic ester and halogenate at the alpha position. Then you take your potassium phthalimide and do an SN2 on there. And then you'd have your N-thaliamido malonic ester that you could then run through and do your reaction to make your amino acid. So this just shows you actually how you'd make that protected amine group. You just do your malonic ester reactions that we talked about in the enolate chapter. Here's another one. This is probably the simplest way. This is also a review of chemistry that we just talked about. This is reductive amination. So if you take pyruvic acid, it is an alpha keto acid. So at this alpha position, it has a ketone. And if you mix it with ammonia, you end up with this imine intermediate. And with that imine intermediate, if you have a reducing reagent around, H-, it will come in and reduce that imine and give you an amino acid. So probably the easiest way to make an amino acid, but you do have to start with a much higher functionalized group. You have to have a carboxylic acid and an alpha ketone group. But this is a review of your reductive amination. You form an imine and reduce the imine. So what's cool about this is it's just a review of chemistry that we've talked about before, but with applications to make biological molecules. Now, in our body, we actually use this reductive amination process to make some of our amino acids, but we use a different reducing reagent because we can't really ingest lithium aluminum hydride or sodium borohydride. So the reducing agent in our body is NADH. And that NADH drops those electrons in there. Those will come around here and kick out this H- that actually acts as the reducing agent. Now you notice what happens in the end is we end up with NAD+, but we end up with an aromatic ring. So a lot of the driving force for this reaction is the aromatization of this purine ring. And anyway, this is how we would do it biologically. And the nice thing in our bodies is they're very good at making a single enantiomer using proteins as the catalyst. Okay, so how do these amino acids actually combine to make proteins? Well, as I said, they just link up the nitrogen to carboxylic acid to make peptide bonds, big long chains of amino acids. And so you can see here, we take glycine, we take alanine, and essentially you're just doing a dehydration reaction. So the nitrogen and the carboxylic acid are going to combine to make an amide. Now, in chemistry that we talked about in the carboxylic acid chapter, we would use an activated carboxylic acid, such as an anhydride or a acid chloride, or maybe even sometimes esters. In our bodies, actually what we use here is there is a transfer RNA. Oh, I can't remember what does a general tRNA look like? Something like that. So you have a tRNA attached, and that ends up being the leaving group when we synthesize proteins in our bodies. So actually that's a little bit of a lie, but that'll work for now. So in the end, we end up with these peptide bonds. And if we combine glycine and alanine, we end up what is referred to as glycylalanine. So we always write out the amino or the peptide chain with the nitrogen on the left from N terminus, the nitrogen terminus, to the carboxylic acid. So that is one reason why I like drawing the amino acids with the nitrogen off to the left, because the convention is we always draw the nitrogen of polypeptides off to the left and the carboxylic acids off to the right. So again, this is what's referred to as the N-terminus, and this is the C-terminus. Okay, well, if we just took two amino acids, alanine and serine, and we just combined them randomly, you'd actually end up with two different dipeptides. You'd end up with an allosyr, or allylserine, or cerylalanine. So you'd actually end up with two different ones. So just from two different amino acids, we can get two different peptides there. Actually, if we did all possibilities, then we should be able to get an alla-alla and a seer-seer. So from two amino acids, you could actually end up with four different dipeptides. So here is just an example of that. If we're only mixing them, so we're limited to non-repeating elements, we could end up with an alla-gly dipeptide or a gly-alla dipeptide. So the peptide linkage is actually somewhat constrained in its rotation, and that's due to this resonance from that nitrogen there. So there is some planarity around this bond here, and that's really important in the structure of proteins because it locks them into certain conformations. We don't want our proteins to be floppy. We want them to have structure. And we know that the nitrogen donates in significantly because we can see it in the IR. It actually shifts the absorption of that C double bond O down into the 1600s, which means that we're actually weakening this bond, and we actually have a significant amount of double bond character there. This would be the greatest contributor, but this does have a significant contribution to proteins and the rotation. So if you look at this little picture at the bottom, around this amide bond, they're actually viewed as kind of being planar, kind of flat. And so what we can do is we can rotate around this bond here, and we can rotate around this bond here, okay? And so that gives rise to our protein structures. There's some other things that give rise to protein structures, and one of those is the cysteine side chain. So thiols, and we talked about this back in alcohol and thiol chapter, but thiols can actually undergo a reaction called oxidation to form what are known as disulfides. And this is a reversible reaction. So under mild conditions, even exposure just to oxygen in the air, thiols will eventually form disulfides. When proteins actually have this thiol side chain, and they're often present in air. And so these sulfides can form disulfides with each other. And in our bodies, they have a very controlled way to form these disulfides. And so what happens is these proteins will fold up into very organized structures held together by this covalent bond from one cysteine to another cysteine. In fact, insulin actually has three disulfide bonds. They have one intra-chain linkage, meaning a disulfide within the same chain. And then we have two interchain linkages. So actually insulin is a dimer of two short peptides. And so you can imagine that you would want these disulfides to be very exact. You wouldn't want this cysteine to form a disulfide with that cysteine, or this cysteine to form a disulfide with that cysteine, and then this cysteine to form a disulfide with that cysteine, or all the other possibilities. That would lead to a different structure and a non-functional protein. So again, in our bodies, we want to control how these disulfides are formed so that we can actually get the correct structure. And there's other molecules that actually have disulfides that are these short peptides. So we got bradykinin, vasopressin, and oxytocin. Bradykinin inhibits the inflammation of tissues. Vasopressin controls blood pressure and regulates smooth muscle contraction. It's involved in the fight or flight hormone. And oxytocin induces labor So it stimulates uterine contraction and also stimulates lactation in nursing mothers. It's a calming hormone. So also often referred to as the snuggling hormone. So if you need love, you need an oxytocin release to help you feel secure and comforted. And you can see that these actually have these intra-chain disulfides. In fact, vasopressin and oxytocin are actually very similar. And you can see that this glycine matches up that proline, that cysteine, asparagine, glutamine, tyrosine, and that cysteine. So they differ, that phenylalanine isoleucine there, and this arginine and leucine right there. So they really only differ by 2 amino acids, but yet have vastly different responses. One makes you want to run away, and one makes you want to cuddle. So here is oxytocin drawn out in all its structural glory. So we can actually see the side chains. And so you can see that this disulfide linkage right here between 2 cysteines actually causes a loop in the molecule. Okay, so there's some other interesting peptides, enkephalins. These are pentapeptides. So these are actually synthesized by the body to control pain, often referred to as our endogenous endorphins. So these bind to natural painkillers. Just happens that synthetic molecules like morphine and Demerol also bind to these receptors as well. So it's thought that morphine and Demerol and these enkephalins actually have very similar 3-dimensional, not necessarily shape, but charge presentation. So if you look at how they look 3-dimensionally, their charges in hydrophobic places would be in very similar spots. And so these bind to the same receptors and have the same response. So if you've ever had a runner's high, that's often due to your body releasing these because you're likely moving into a pain phase and your body's trying to mitigate that. And if your body can't produce enough of these endorphins, then, or the pain's too much to bear, then you use these synthetic derivatives that again bind to the natural receptors. Here's an interesting dipeptide. This is called NutraSweet or aspartame. And so it contains aspartic acid and phenylalanine. And so it's a non-nutritive sweetener at 200 times sweeter than glucose. And some interesting things about it, this methyl ester is sweeter than an ethyl ester. And actually, if you change these to D-amino acids there, it actually tastes bitter. If you have any compounds that have NutraSweet as a sweetener, you'll notice that there is a warning on the side, phenylketonurics, and it contains phenylalanine. So some people have PKU where they can't metabolize phenylalanine efficiently, and so if they eat proteins, foods high in phenylalanine, this will collect in their brain and end up with mental retardation. So proteins, as I said, have shape and structure, and we refer to this as the primary structure of a protein, secondary structure. Primary structure is just a sequence of amino acids, but the secondary structure are the way these chains of amino acids actually fold up. And so one of the ways they fold up is into an alpha helix. And you can see it here with the atoms shown in a ball and stick model, and also in what they refer to as cartoons, where this coil here is representing an alpha helix. And how this works is you get a hydrogen bond from the carbonyl on one amino acid to the nitrogen on or the amide nitrogen hydrogen bond on another amino acid. And so it coils up. You can also get hydrogen bonds from one chain to another. It actually can be the same chain, but essentially it's not just one that's up the chain, it's actually across. And so you can see that these hydrogen bonds line up neatly. This was the secondary structure discovered, and so they refer to it as the beta sheet, where the first one discovered was the alpha helix. So beta sheets can actually coil up into these pores, or these beta barrels. And right here is actually a modified tyrosine amino acid. And this protein right here that just has this beta barrel actually fluoresces green. And so they use it in molecular biology quite frequently, and they get animals to actually express this protein. And then when you shine a ultraviolet light on it, they fluoresce green. They've modified it too so they could actually fluoresce other colors. So these are actually animals that are transgenic. They actually have this jellyfish is where they first discovered it, this jellyfish protein in them. And so you can see here, these naked mice are expressing the green fluorescent protein where these mice are not. And you can do it in fish and all sorts of organisms, from animals to insects to fish to nematodes to cats. And these are pigs. Okay, so protein secondary structure is how these chains of amino acids coil up or align to interact with each other. And so, as I said, we actually have two that we've talked about, these alpha helices, or here, these beta sheets. Now, there's lots of ways to keep a protein in shape. Besides these secondary structures, there are a bunch of other interactions that can occur between the side chains. And so when we look at the overall structure of a protein, we refer to that as the tertiary structure. These tertiary structures include alpha helices and beta sheets, but they also include turns and kinks and bends and all sorts of things. So there's lots of interactions from one side chain to the other, such as just hydrogen bond interactions, as we see here. These are all hydrogen bond interactions. You can get hydrophobic interactions. So these non-polar side chains like to be in proximity to each other. They don't like to be near other polar side chains. So like dissolves like, or like side chains like to be next to like side chains. You can get what they call salt bridges, which is the interaction between the negative of a carboxylic acid and the positive of an amine group. Or you can get these covalent interactions here, disulfide bridges. So these all are ways in which proteins interact within a structure to make a specific protein shape, which we refer to as the tertiary structure. And then some proteins are actually multi-subunited. So they have multiple subunits, such as hemoglobin, which is this structure here. This is what carries oxygen. So hemoglobin is a tetramer of two identical subunits. And so we have these green subunits here. We have these orange subunits. So altogether, there's 4 proteins that make up hemoglobin, the functional protein structure. And that is what's referred to as quaternary structure. So it's a 3D arrangement of individual peptides within a multi-subunit protein. So this has multiple subunits in it. And so here's an example of a hexamer. So we've got a couple different ways that we could arrange that hexamer. And so you can see that by including other subunits, you can actually vary the type of overall protein that you get, and therefore vary the structure. So there you go. There's a summary of the type of protein structures. We have the primary structure, which is just the sequence of the amino acids. We have the secondary structure, which is how the side chains interact to form these structures such as alpha helices and beta sheets. We have tertiary structures, which is how the whole chain folds up into a structure. And then in some cases, we actually get multiple subunits that interact together that give us a quaternary structure. And we'll leave it at that. So we'll have a brief lecture on lipids, and we'll be done with biomolecules.